Welcome students to the next session of the lesson Strategies for Enhancement in Food Production. Today we will be dealing with some technology-based methods of animal breeding, artificial insemination and multiple ovulation embryo transfer. Let me first recap with the topics already dealt with. We discussed various methods of animal breeding naturally. It is under controlled breeding experiments. We discussed inbreeding and outbreeding, and a breeder could do it naturally mate superior males and superior females of his farm for better progeny, so the yield would be better. We discussed in detail about outbreeding methods, outcrossing, outbreeding and interspecific breeding. I would like to mention here that crossbreeding example, because I was dealing with cattle, I explained with examples of two different breeds of cattle. Among cattle, I discussed cow, cross of cow and a bull of two different breeds. But I would just tell you, please go through this example of sheep, because this is very important as given in the NCERT. Hizzardale is a new breed of sheep which is a product obtained by crossing Bikaneri Eves and Merino Rams. This is very important, though it is just said in one line as an example, but please learn this example. Hizzardale is the new breed, and the female here taken is Bikaneri. Eve is a female, and Merino Rams. So you can remember by RAM here, MAR. Backward RAM. So Merino is the male, Ram, male. Okay, so Merino Rams. So that way you can just remember this for examination point of view. So these were the methods we discussed what breeders generally do in their farm. But the yield is not up to the mark, so better technologies have come up. So we'll deal with some technology. 1. Artificial insemination. The word itself tells you that this is done with assistance by human beings, artificial. It's not the natural mating process in animals. Insemination, that means there is an assistant. The breeder allows that inseminating process in the superior female. So for that, it is very necessary that you collect semen. So what is artificial insemination? It is a method of carrying out desirable breeding among animals. That means you choose the male and female again here. And this is not done through natural mating. This is done by collecting semen from a superior male. And then it is injected into the reproductive tract of the selected female by the breeder or any expert person in the farm. So see the technique. The semen is collected. We get frozen semen. Frozen semen is available in cryopreservation centers, semen banks. They are preserved by a method called cryopreservation, deep freezing method at -196 degrees Celsius. So semens are available. You can get semen from, the breeder can get semen from anywhere. He can order for it, and he may get a vial of semen which he can even store. Suppose he is not using all the semen which he obtains in one vial, he can use it for later purpose. He can store it in this frozen manner. So see the technique. Through a device, it is inseminated. It is given into the right area, reproductive tract of the female, which is ready. The reproductive cycle is monitored and the female ready in that phase where ovulation could be possible. So at that period it could not be given any day, but the cycle is monitored and the semen can be given into the tract. So this method is called artificial insemination. Frozen, collected and given into the cow. Okay. Now, what could be the normal problems? Why should a breeder go for artificial insemination? There are many problems with normal mating in animals that a breeder faces. Suppose he is not having a superior quality bull with high genetic merit. So he has to get the transportation of a superior bull done from some other place to where his superior cow is housed. So that is a very tedious job. Then natural mating, there is a lot of wastage of semen. Then there is no way of collecting healthy sperms. There is no option for choosing. It's a natural process. So time is wasted. You have to wait for the progeny. Then you have to evaluate the progeny, whether they are having the good gene in them, whether they are of high quality, where their product would be good. It takes a lot of time monitoring all this. Then even conception rate is very low. It is not necessary that in normal mating, even if you suppose transported a superior bull and allowed mating with the female cow in your farm, there is no guarantee that this female will get impregnated and she will give birth to the calf. So that conception rate, conceiving, is not guaranteed here. Chances of spread of diseases is also there. You cannot take care of the hygiene manner of allowing mating and all. So all those problems are there. So artificial insemination is a technique that overcomes problems of normal mating in cattle. To some extent it solves the problem. That is, you need not have any transportation done for this, because as we discussed in the previous screen, you can have semen available. You can just order for it. Semen from the desirable cattle can be obtained from any semen bank. You can even choose the breed you want. Then the banks select and preserve healthy sperms. When semen is collected from bull, they even do the selection process of only keeping the viable healthy sperms. So that is good. Semen ejaculated once from one superior male that you get in a vial may be millions. The sperms in them may be millions. You can use it for inseminating hundreds of female cattle. Suppose your farm is not big enough. Suppose you have 25 cows which are in the reproductive age. So after using for those 25, you can still keep it away for more. So that way you can use hundreds and hundreds of female cats. You can even give it to some other farm who may be in your contact. Okay, so no semen wastage is there. You can use it to the maximum. Better conception rate. The female generally, suppose you have inseminated the semen to suppose 20 cows. So good chances that more number, at least 7 to 10 cows will definitely conceive. So that guarantee is there, better than the natural method. Then the process carried out is with very good care. Hygiene is taken care of, utmost care and hygiene. So chances of diseases are very less. So all these are the main things about the two technique and how artificial insemination is better. It overcomes some problems of normal mating. But again, success rate of the production, that is the hybrids produced, the progeny produced, and the yield from them is still not up to the mark. That means the ever-growing demand of food or milk and milk products or anything, any product that you are rearing the animal for is not up to the mark. So that means AI, artificial insemination, does not meet that demand as we need. So there are still higher techniques. The next level of breeding technology is called MOET, multiple ovulation embryo transfer. This technology, MOET in short, very important for your lesson. So what we do here is a genetically superior female, that means of a good gene of high lactation, whatever we discussed in detail about superior female, qualities of a superior female, that is taken care of. Then when the reproductive period is understood of this cow, at the right time, that means when follicles are to be developed, at that time FSH, follicle stimulating hormone, is administered. That means the cow is injected with this hormone. See, in brief I'll tell you, this FSH hormone is a very naturally produced hormone in the body of all the mammals, that is from the pituitary, and normal secretion of FSH triggers ovulation, triggers the process of follicle development. So normally a female develops, like cows, humans, etc., they develop only one egg, that is one follicle develops into a mature ovum. a time in one cycle. That is not enough if you have to improve the herd size in your farm. You cannot wait for every time one calf being produced. So this process helps. FSH, overdose, is given to the cow. So instead of one egg, one follicle developing into a mature egg, many follicles develop. Here I am showing you what is happening in the reproductive tract, in the ovaries here. That means development of the follicles. So instead of one, this is one follicle, this is another developed, this is again. So all these are follicles. So many number of follicles would be developed at a time, which is not possible in a normal cycle. Okay, so that is because of follicle stimulating hormone. Now, when these eggs are developed, the female, when it ovulates, that means developed follicle will rupture and the ovum would come out, oocyte will come out into the reproductive tract, fallopian tube, where fertilization occurs. So at this time, during ovulation, if this animal, that is the cow that you have selected, is naturally mated with an elite bull, elite, high quality, high class bull, or second method could be you have semen collected from some superior bull. Any of this can be practiced here. You either allow the bull to naturally mate with the cow during the ovulation time, or you inseminate this cow with the semen you have collected. Clear? So what happens if when this is done, naturally fertilization chances are very high. Again, this is shown out. This is happening in the reproductive fallopian tube tract. So many millions of sperms, I have shown only a few, millions of sperms would be there in a few micro liter of this thing. So what happens is these sperms would fertilize many of the follicles which are ready. Good chances are that most of them would be fertilized. So see the internal structure of the cow here, again the same cow. So what happens is when this is fertilized, next step, zygote would be formed. When zygote develops, all those fertilized eggs should be allowed to divide through mitosis and reach up to a stage of 8 to 32 celled stage. This is magnified view of what has happened in the internal tract. Okay, so all the fertilized cells, that is zygotes, which have reached 8 to 32 cell stage, can be retrieved through a catheter. You can retrieve it from the body of this animal through a catheter. Okay, collect it, and non-surgically. You need not use any surgical methods. You just use this catheter. Here is the device picture. And now there are so many other cows in this farm. I have drawn only 3, ready to receive these embryos. Their reproductive cycle is also monitored. Their uterus is ready at that phase. They are synchronized. Their reproductive cycle is synchronized. So these eggs one by one can be given into these animals. Only one at a time, because only one calf would be born. So from this cattle, it is retrieved and given to this surrogate mothers. They will have, the gestation period will be in this cattle, and they will give rise to, give birth to the new progeny. Clear? The young ones born are genetically similar to this cow. They have the gene of this mother. This is a genetic mother. These are only the surrogate mothers. This is done to improve the herd size. This female cow is not going to give birth. She is free after giving away the embryos in this stage. She is going to be ready for the next cycle of superovulation by administration of FSH after a period of some weeks. I hope the topic is clear. So this is a good method of increasing the herd size. Any doubt, you can write in the comment box. Thank you so much.